I'm going to actually go ahead and make a start. We'll keep our fingers crossed that the powers of modern technology will in fact succeed, and we'll bring in our next speaker remotely. And so while we're working to do that, I just want to introduce the context of this afternoon's session. As I listened to this morning, I think the strength of the morning was the overall science that we heard and the quality and the advances that have been made in the biology of tissues on chips. I think the inherent weakness of this morning's session was there simply wasn't enough time for questions and discussion. And so, fortunately, I think the organizers figured out that that was going to happen. So, if you will, this afternoon's session is designed to specifically turn those attributes on their head, as it were, and hopefully what we have is a very dynamic and very interactive session. So, what we're going to try to do this afternoon, based on what you've heard this morning, as well as a few presentations that we'll have, is ultimately lead to a discussion where we can really explore the science and focus on the kinds of challenges that we see that will be necessary to achieve the next steps in how we apply tissue chip technology to environmental health in general. One of the things that I heard this morning was someone raised the point that with some of our systems we can ask better questions. I would actually turn that around and say, I think, if you will, think about, for this afternoon's discussion, whether or not we're getting better answers by using these systems as we approach the science. So with that as a very general introduction to the scope of what we're going to do, I'm going to hope and pray that's modern technology, that we have Francisca Bose on the line, who's going to present remotely for us on the comparative assessment of 3D liver models for predictive toxicology.
So, yes, let's hope you can hear me. Maybe quick feedback, is it okay?
Yes, we can hear you.
Great. So, first of all, thank you very much for giving from pharmaceutical industry the opportunity to share their views and experiences when it comes to the application of more sophisticated organ models. As the title already says, this presentation will almost exclusively deal with liver models, as that is where we have our expertise in this group. So, oops, slide's not moving, just a moment. And I hear a terrible echo, so I switched the speaker off. Please keep questions to the end of the session. So I hope it's better now. So let me quickly introduce the status of vitro toxicology in our company as of today. Where we still, in vitro methods in toxicology are currently mostly applied in early phases of drug development or already in drug discovery. It's about exclusion of drug candidates with favorable safety properties early, before considerable effort has been invested, expenditure of resources, such as time and money, and animals have to fail cheap is the motive. So industry has adopted methods to address that's undesired. So for example, it's about metabolic induction or enzyme inhibition, genetic toxicology such as mutagenicity or clastogenicity, cardiac function, here the famous Herc assay, or teratogenicity. These are all well-established assays, validated assays. Where we still struggle and what implies a major organ toxicity. Organ toxicity that we heard this morning already, often complex and often the result of mode of action, but a combination of several effects. Such a multitude of factors are involved, and by and large, this is sort of an interplay of all types. Often, these toxicities also so it's very difficult to address. It involves complex and dynamics of
To unmute your line, press *6 or #6.
Case detection via simple screening tools. The pie chart illustrates in this slide the contribution of different organ toxicity in clinical failure. And you can easily spot that toxicity to the cardiovascular system and the liver are the two major causes of failure of drugs in clinics due to organ toxicity. This, of course, explains the high interest of industry in efforts into these organ systems. Within the Roche, the Mechanistic Toxicology group I reside in deals with organ toxicity at different levels of the R&D process. From the very early stage of target assessment on the theoretical level, depicted on the very left, over assessment of specific modes of action such as mitochondrial toxicity, direct toxicity to bone marrow stem cells, or interaction of compounds with bile salt export pump as risk factors for liver toxicity, depicted in the box in the middle, up to the assessment of the causes and modes of action for toxicity observed in animal toxicity studies, depicted in the box on the right. Here the most frequent question is the relevance of a specific finding in animals for humans, which can be addressed by finding out more about the mode of action and comparison among species in vitro. Learning from these later investigations, especially learning on the mode of action of a certain compound causing toxicity, can then, of course, be fed back to early compound prioritization with assays only applicable to a certain compound series and used for compound prioritization in this specific project. Learning from the investigative part, however, can even lead in the establishment of predictive assays, applicable more broadly and rigorously validated as already mentioned. Using in vitro models for work done in this right box, we call this issue resolution and de-risking work, has the unique advantage that the in vitro system you intend to use for this work can be qualified with respect to their suitability by comparing to in vivo. And here, often experiences that certain in vitro models are not suited, while others, maybe more complex ones, and not necessarily always the same to be said, are able to recapitulate certain features of this in vivo finding and can thus be used to further elucidate aspects of the mechanism of action. With the following case example, I'd like to illustrate the advantage of a more complex 3D model compared to more simple 2D systems and illustrate what just said. In this case example, neoplastic changes were observed in livers of rats upon 26-week treatment with one of our development compounds. No comparable changes were seen in dogs upon comparable exposure duration, so also 26 weeks. From short-term studies in rats and additional ex vivo investigations, we learned that very early on, i.e. on day 1 only, the compound induced metabolic changes in hepatocytes characterized by glycogen accumulation, shown in the P and D stain at the bottom, as well as proliferation, seen as an increase in Ki-67 positive cells. Ki-67 increase was only seen on day 1, not later on, and I hope you can spot that from the bar graph depicted in this slide. In addition, by ex vivo liver gene expression analysis, we learned that the mode of action seemed to be distinct from the well-known rodent-specific neoplastic changes induced by nuclear receptors CAR and or PXR. The request to our team was to elucidate the mechanism of action and to come up with an assessment of the relevance of these findings for humans, of course. As proliferation was the finding that was most easily to be linked to the pre-neoplastic changes, proliferation was deemed the most important phenotypic endpoint, especially by the project team. Unfortunately, we were not able to recapitulate proliferation, as seen in rats in vivo in a simple monolayer rat hepatocyte culture system. Thus, we also tried to recapitulate proliferation in co-cultures of hepatocytes and non-parenchymal cells, as we learned from earlier work with especially the PPARs that proliferation with PPAR agonists is more pronounced and more reproducibly seen in hepatocytes when non-parenchymal cells are present in the cultures. The graph you see here depicts the results obtained in this co-culture model and shows that there were only single cultures, single batches of cells which reacted at all to the positive control proliferation inducer recombinant human EGF. In these batches, also our test compound responded, but in most of the cultures, nothing happened. And of course, this is not a system you can use for doing species comparison. However, when we used a more complex 3D multi-cell type liver culture system for our investigations, we saw not only a reproducible proliferative response with the positive control inducer EGF, but also with our development compound. The graph on the top shows the results of 3 independent experiments with 3 different batches of primary human cells. And on the bottom graph, you see the results of the species comparison when summarizing 3 independent experiments, each for the 2 species. It shows in the right panel that the human system reacts even more pronounced to human EGF as the positive control than to the rat cells. However, there is no increase in response to our Roche development compound in human cells, while there clearly is in rat cells. In summary, the proliferative response induced by our Roche compound in rat liver could only be recapitulated in a more complex 3D liver system. And the species comparison in this in vitro system strongly suggests a species difference. The 3D multi-cell type liver culture system we used in this example was developed and commercialized by Dawn Applegate and her company Regenemed. In our lab, the system was intensively characterized and the respective results published by Radina Kostadinova. The culture is formed by co-culturing primary, expanded, non-parenchymal cells, first alone on 2 interconnected nylon meshes, serving as a scaffold. The non-parenchymal cells are cultured first for 7 days. Only then, when the cells have already produced some extracellular matrix, as can see in the photograph on the bottom right, the hepatocyte cell fraction is brought into the system. After another at least a week, the tissue formed looks on a histological level as depicted in the photomicrograph at the bottom right. And you see the different cell types enwrapping the fibers of the nylon mesh. The culture system is designed as a Transwell system and comes in different formats from 6 to 96 well plates. Our characterization work revealed albumin, transferrin, and fibrinogen secretion, depicted in the graph on the top left, as well as urea secretion, depicted on the right, being considerably higher than in 2D cultured hepatocytes and remaining relatively constant over at least 10 weeks in culture. As illustrated in the graph on the bottom, glycogen production in this culture system could be increased by insulin treatment. Still, after 2 months in culture, the system reacted with the production of IL-1 beta, TNF-alpha, GM-CSF, IL-6, and IL-8, the 2 depicted on the left, to stimulation with LPS. This finding is indicative of the presence and functionality of non-parenchymal cells, we think. The graph on the right shows that cytochrome P450 activities, here CYP3A4 activity as an example, could be measured at more or less stable level over the course of 3 months in culture. Moreover, the enzyme activity was inducible with respective inducers and inhibitable using respective inhibitors of the enzymatic activity. Moreover, when trying to measure the metabolic activity of the system with some in-house compounds with less nice physical-chemical properties, disappearance of the parent compound could be observed, while respective metabolites were not retrievable. This indicates that unspecific binding, most probably to the material of the culture scaffold, may constitute a considerable problem of this culture system and maybe of other systems with scaffolds and large surface areas too. When comparing the toxicity of certain model compounds in the 3D system to conventionally cultured hepatocytes in 2D culture, considerable differences were observed for certain compounds. Depicted here is troglitazone. This is a compound withdrawal from the market due to idiosyncratic liver toxicity about 2 years after its introduction. While troglitazone shows some toxicity in rat but not human 2D cultured hepatocytes, this is depicted on the, right? The picture in the 3D systems is vice versa. Cytotoxicity is observed already starting from the first day of treatment in the human 3D cultures at concentrations of about 7 times the in vivo reported plasma Cmax. That's on the bottom left. No toxicity is seen in rat 3D cultures up to the highest concentrations. As another example, trovafloxacin, an antibacterial drug of the class of fluoroquinolones, was removed from the market due to the induction of hepatotoxicity, was compared to another drug of the class, levofloxacin, which is currently still thought to be non-hepatotoxic. In the human liver culture system, trovafloxacin revealed some cytotoxicity in vitro. As can be seen from the top left panel, toxicity became overt with concentrations of about 3 to 4 times in vivo plasma Cmax, but it was only seen after repeated treatment of the culture system for more than a week. The different line colors indicate LDH release at different days in culture, and the red line, that's the one showing the most pronounced increase in LDH release with higher concentrations, depicts the data from treatment day 8. No toxicity was seen with levofloxacin on the bottom, despite treatment with concentrations covering up to 10-fold in vivo plasma Cmax. From the panel to the right, it can be seen that no toxicity of either trova or levofloxacin was seen in 2D cultured human hepatocytes, which, due to the nature of this short-lived culture system, the 2D hepatocytes were only treated for 2 times 24 hours. That has to be said, of course. Thus, in summary, we found that the performance of this 3D culture system is very promising. In our hands, this culture system showed long-term stability, some drug metabolism activity, seemed to retain some immune-competent cells functionality, and which, important for our issue-driven and species comparison approaches, the system was able for different animal tox is available for different animal tox species. And that's, in our case, coming from the industry and working in vivo with animals, very important to always compare to, let's say, the in vivo issue. The drawback of the system is, as for us here in Europe, the cumbersome logistics. So Regenemed is sitting in San Diego, as well as some quality issues with the scaffolds. In addition, we of course did not like the fact that we were not able to retrieve metabolites of several of our test compounds. As already illustrated with the example of the pre-neoplastic liver foci-induced development compound, we were, however, confident enough to apply this culture system for some of our investigative work on projects. From a practical aspect, however, we did not deem this system suited for a more routine application to all projects, or to being a tool to screen for hepatotoxicity with a reasonable throughput. As already mentioned, the fact of not being able to retrieve metabolites from the culture system despite the disappearance of parent compound led us to suppose a problem related to non-specific binding of some of our compounds to the scaffold material. Non-specific binding is, however, not a particular problem of the shown 3D system, but may occur with many other systems as well. And as already mentioned, this might be especially a problem with scaffold systems or systems with tubing, devices with high culture, device material surface. What you see here is investigations done by some of our DMPK colleagues around nickel crotocryl. They tested non-specific binding in a microfluidic system called the Celasix Pearl system. You can see that depicted on the right. In this system, hepatocytes are aligned in channel with pores, and the medium containing test compound is flowing in between in attractance channels. The system comprises of 3 units, the size of approximately a well of a 96 well plate. So the medium containing test compound is loaded into the inlet well indicated by the red arrow, and flows driven by hydrostatic pressure through the microfluidic chamber loaded with hepatocytes. You can see that in the middle, and flows then to the outlet chamber, indicated with the green arrow. The numbers given in the table show that test compound concentrations in the outlet wells, and sometimes even in the inlet wells, have markedly decreased upon 24 hours incubation, and that is in the absence of any cells in the device. It is indicative of high non-specific binding of test compounds to the material of the cell culture device, of course. As already explained, the Regenemed 3D system in our views is not suited for more routine application. That is why we looked into other cell systems to be applied to the more prospective, predictive part of our work, i.e. trying to predict in vivo liver toxicity by the use of in vitro systems. During the testing of various other liver in vitro systems, we, like others, showed that exposure time matters, of course. Depicted here is the flagging of compounds as being hepatotoxic or not in 2 culture systems amenable to higher throughput assays. Flagging is based on the occurrence of effects on one of several measured parameters below a certain threshold concentration. This threshold concentration is set to several times the in vivo Cmax of the compound. The 2 systems depicted here are the heterogeneous hepatopack culture system, a micro-patterned hepatocyte fibroblast culture system, and spheroids of hepatocytes formed with the hanging drop method. The in vivo liver toxicity classification of the compounds used for the assessment can be found in the first colored column to the left, red meaning in vitro liver toxic, green indicating non-toxic. For each culture system, the toxic or non-toxic outcome upon 2 days repeated test compound treatment is depicted in the more left column. In the right column, results are depicted upon 5 repeated treatments. And not surprisingly, it can be seen that the coloring, the matching with the in vivo color coding is higher in both culture systems with increasing treatment duration. And thus, hepatotoxicity prediction accuracy is increased. That cytotoxic concentrations decreases with repeated treatment has already been shown and published by others, and as said, sounds relatively obviously. For example, using the Hepatopack micropatterned culture system, Kedani and co-workers have shown that the predictivity for hepatotoxicity increases upon 9 days treatment compared to conventional 2D hepatocyte culture treatment for 24 hours. So something very similar as we have done. The color table depicts the results obtained with the Hepatopac system upon 9 days dosing in the most right column for 25 compounds wrongly predicted with the 2D system. I think this again emphasizes the importance of repeated and longer-term treatment in vitro. In the Hepatopac culture system, the hepatocytes are figuratively spoken cultured as islands of defined size and shape and with defined spacing in a sea of 3T3 fibroblasts. This configuration has been shown to support good hepatocyte viability over weeks in culture, as indicated by high albumin and urea secretion. In addition, the system quite well retains some xenobiotic metabolic function, especially the P450 mediated phase 1 metabolism. I think therefore this system is currently establishing as the best suited model to estimate clearance rates and half-life of low clearance compounds in the pharmaceutical industry. The system is very nice to work with from a practical point of view, as it can be handled like conventional cultures and is amenable to automation type content imaging. Somewhat non-physiological, of course, and also making things a bit more complicated with regards to results interpretation is the presence of the large number of fibroblasts, which are of non-liver origin, and even from a different species, namely the mouse. Moreover, lifespan of the culture system is considerably shortened under serum-free culture conditions, as serum is needed to support the longer-term survival of the fibroblasts. Up to recently, the lack of the presence of non-parenchymal liver cell types in this system was limiting the use in our group, as these cell types are thought to contribute to or at least modulate liver toxicity. However, it has to be said that recently the company launched a system with immune cells. As said, because we regard the presence of non-parenchymal cells as very important in liver toxicity prediction, we decided to have a closer look at liver microspheroid systems containing non-parenchymal cells. The microspheroids, or microtissues, how they are called by the producer company Inflammatory, resided here in Switzerland, are formed by the hanging drop method and then spun down into specially formed conical wells of a 96-well culture plate. The picture on the top right shows a microtissue in one of these wells with a bottom diameter of 1 millimeter, so one can easily see that the size of the microtissue is about 200 to 300 microns. The microtissues comprise of about 3,000 cells in total and have been shown to contain liver non-parenchymal cells, as shown in the bottom right picture for copper cells. This picture shows immunohistochemistry staining of a microtissue for CD68 and macrophage cell marker. So this darker brown spot clearly shows the presence of copper cells. In our hands, the Infero microtissue culture system has shown good viability with albumin and urea production in a close to physiological range over several weeks in culture. It has shown retention of some basal as well as inducible metabolic, i.e. P450 function, and it has shown cytokine release upon LPR treatment, again emphasizing the presence and functionality of immunocompetent cells. Liver microtissues are available ready to use in 96 or 384 well plates for human as well as for the most common tox species, again important for us. And from the practical aspects, the system is easy to handle and would even allow culturing, automation of culturing. Drawbacks of the system might be the low number of cells per tissue. This increases the requirements with regards to sensitivity for the readouts of interest. Furthermore, the relatively dense structure of these microtissues may limit the speed of perfusion of nutrients, test compounds, and staining dyes. And last but not least, the imaging of real 3D construct like this is more challenging than for systems comprising of only one single cell layer. With regards to the biological performance, results obtained with this culture system were quite similar to what we observed with the scaffold system presented before, the Regenemed scaffold system. The table on top shows IC50 values in micromolar for some model compounds in conventional rat and human 2D culture, and the Infero microtissue culture system. Again, it can be seen that for some of the compounds, here amodiaquine and troafloxacin, the IC50 is lower in the microtissue system compared to the conventional 2D hepatocytes, and this may again be due to prolonged, in this case, 8-day treatment duration, compared to only 48 hours in the conventional 2D monolayer. Of note is the differentiation of troafloxacin and levofloxacin, marked with this green frame. So again, we saw some toxicity with troafloxacin, but not levofloxacin. And also very encouraging is the marked species difference for troglycerol toxicity, marked with the red frame. These data are very similar as the data obtained with the Regenemed system, where for rats, the 3D system seems to be less sensitive than the 2D system, while for humans, the 3D system is more sensitive than the 2D system. So as a comparator, I once again depicted the results obtained with the Regenemed culture system on the bottom. But all in all, our first comparative results with these microspheroids, I think they look pretty promising. So you may have noticed that most of the results shown so far were of qualitative nature, comparing liver cell functions in various culture systems to one another, or comparing the toxicity of comparative compounds in the various systems. In that respect, several of the assessed more complex liver culture systems look really to be promising in our view. Thus, it seems that improved physiological parameters in these 3D systems do translate into better, I would say, qualitative in vitro to in vivo correlation. There are, of course, a couple of challenges, as already mentioned earlier by Lance, that have to be addressed, especially with regards to applicability of the culture system more routinely in regular testing in the pharmaceutical industry. These are about ease of use, throughput, technical problems such as non-specific compound binding, as well as reproducibility. This is always an issue when working with primary cells of human origin. But here, we, as others, set, of course, some hope in the development of differentiated cells from iPS. Although the current more complex, more physiological 3D in vitro models reveal effects in concentration ranges more closely resembling the measured in vivo concentrations, I personally think that prospective quantitative prediction remains a big challenge to be solved. Predicting from an in vitro system at which plasma concentrations corresponding effects will be seen in vivo is by far not possible with the systems and methods we have today. Thus, there is a need for even more physiological models and the incorporation of pharmacokinetic principles as well as readouts into these models. So the inclusion as flow, as we heard already this morning, compartmentalization, difficult word, and in the long run, the concentration of different tissue models may provide solutions here. Several examples and the ultimate goal of the human-on-a-chip approach have, of course, been extensively touched upon already this morning. So, however, it goes without saying that the challenges, especially with regards to high non-specific compound binding and practicability, will remain the same, or will even gain on importance with the development of even more complex models, two of which are depicted here. For the time being, and most possibly also for the future, we should, however, not forget that some specific aspects and parameters can be obtained with rather simple systems, and that modeling and simulation is of great help and importance in bridging some of these single aspects together and interconnecting them to model more complex events such as the PK behavior of drugs. So while working on a more physiologicalistic organ models, we should not reduce our efforts to improve also our more simple system and our modeling capabilities. And we should not forget that sometimes comparative or qualitative data are good enough to guide our decisions. Thus, with emphasizing that ultimately the application will define the suitability of various in vitro cell models, I like to close, and I, of course, like to thank all the people who worked with us together on these various in vitro liver model systems showed here, and I, of course, would like to thank you very much for your attention, and I hope, despite the problems with echo and the line, it was not too tiring to follow this presentation by webcast and right after lunch break. So thank you very much, and I'm open for questions if I hear them.
Thanks, Francisco. Actually, you came through loud and clear. So there is at least one question, and we'll allow a little time here. So please identify yourself and definitely use the mic.
Hi, I'm Ivan Grusin. I'm a member of the committee. Francisca, thank you so much for making this presentation. I wanted to follow up on the challenges that you brought up in terms of the practical use of these more advanced systems at Roche. If you can perhaps, give us a relative percentage of compounds going through various liver models in vitro, from traditional 2D cultures to more complex 3D cultures. Can you tell us what is the relative percentage of your compounds now still going through traditional 2D versus the novel 3D?
Yes, so most of the compounds go to the traditional 2D and even simpler systems for mechanism of action, such as mitochondrial toxicity, and these more complicated 3D models are used for issue resolution, as I already explained. Let's say one of 20 where we know that we have a leading compound or so.